Okay, let's take a look at inheritance. First, let's look at chromosomes. Before the cell divides within the nucleus, a number of characteristic thread-like structures gradually become visible. Those are the chromatin. These are known as the chromosomes because they stain intensely with particular stains. The number of chromosomes is characteristic for a species. Once the chromosomes are arranged, the diagram that is made is known as a karyogram. Some of the characteristics that can be taken into consideration when forming karyotypes are each chromosome has another matching chromosome, making it a homologous pair. Each pair of chromosomes is given a specific number. In humans, remember there are 22 autosome pairs, and then one last pair, which would make the 23rd pair, are the sex cells. The homologous pairs can be distinguished since they have distinctive banding patterns when they're stained. One pair of chromosomes, the sex chromosomes, are displayed to one side so that the sex of the organism can be determined quickly. The other pairs are known as autosomes. They're also known as somatic cells. Each chromosome has a characteristic set of genes which codes for different features on the organism. This is showing you a karyogram where you have, for a human, you have the 22 pairs of homologous chromosomes matched up, and then that last 23rd pair is the XY chromosome. So in this case, since it's an X and a Y, we're looking at a karyogram for a male. If it had been two X chromosomes, that would have been a female. Gametes and reproduction. Animals and many plants reproduce by sexual reproduction. Sexual reproduction involves the fusion of gametes. Gametes are the egg and sperm cell. When an egg and sperm cell combine together, that's known as fertilization. And then the fusion of those two gametes is going to produce a zygote. The zygote will then begin going through mitosis to produce a multicellular organism. The gametes are going to be haploid. Haploid meaning half the number of chromosomes that the organism has, which is important to have so that when the gametes fuse together, the organism will have one complete set of chromosomes. The cells of organisms usually contain two sets of chromosomes. These cells are said to be diploid, which is represented by 2N. The N represents the number of chromosomes in one set. Red blood cells are an exception because they do not have a nucleus. Red blood cells are formed in the red bone marrow of bones. The stem cells in red bone marrow are called hemocytoblasts. They're going to give rise to the red blood cells. Human body cells are considered to be diploid, containing 46 chromosomes. However, some cells contain only one set of chromosomes. They are said to be haploid. Think haploid, half the number of chromosomes than the organism contains. They are represented by N. Human gametes are considered to be haploid, with only 23 chromosomes. The number of chromosomes in a complete set varies for different species. Again, in humans, haploid is 23. Mosquitoes would be haploid 3 chromosomes, and in an avocado tree, they have 12 chromosomes. Homologous chromosomes, these are two sets of chromosomes taken from a human cell. Their individual photographs have been moved around so that the chromosomes are arranged in their matching pairs. So as you look at the karyogram and you're looking at the numbers 1, 2, 3, that's representing, like 1 is representing one pair of homologous chromosomes. 2 represents the second set of homologous chromosomes. Homologous chromosomes are the same size, they carry the same genes in the same position. That position is known as the locus or the loci. Doesn't mean that the genes have to be the same. It would be the same gene but a different allele. Meaning you could have a location, we'll look at this again, but you could have, let's say this little location right here, the loci right here, is representing eye color. Then eye color would also be represented on the homologous chromosome at the same loci. This one, however, could be blue eyes. This one could be brown eyes. So the gene is the same, but the allele could be different. Okay, question time for you. If you would please take a moment, pause the video, and answer these 2 questions. Taking a look at the structure of chromosomes, they have their distinct structure. They look like they have a double structure. Each chromosome is made up of 2 identical structures known as sister chromatids. So this represents one chromatid. This is the sister chromatid. The sister chromatids are attached at the centromere. Remember, the centromere does not need to be in the center. This occurs because during interphase, each DNA molecule in the nucleus makes an identical copy of itself. Each copy is contained in a chromatid, and both are held together by a narrow region known as the centromere. The position of the centromere is characteristic for a particular chromosome. Since each gene is one unit of inheritance, the gene for a particular characteristic is always found at the same position, or the locus or the loci, on the chromosome. So if you're looking at chromosomes, this one's not duplicated yet. So here you're looking at different genes. Okay, these bands are known as the different genes. When we have the duplicated chromosome, you have the sister chromatids. It's going to duplicate those bands. So the genes would also be duplicated. And then again, the sister chromatids are going to be attached at the centromere. Looking at homologous chromosomes. The members of each homologous pair of chromosomes comes from one of the parents. In humans, 23 are maternal chromosomes and 23 are paternal, meaning mom and dad. Each pair contains genes for the same characteristics, but they differ in the way that these are going to be coded for. Again, the gene would be the same, but the alleles would be expressed differently. Mutated alleles may code for a particular characteristic that is faulty, such as a particular disease. And that's what your diagram is showing you, how that mutated gene could get duplicated and then passed on. Looking at an example using the fruit fly. Fruit fly has 5 pairs of chromosomes. If we take a look at the chromosome, this first one is showing you the location for 4 genes on a chromosome. You've got one that's representing the length of the appendages on the head, determining the body color, the wing length, and then the eye color. When it's with the homologous chromosomes, again, the genes would be the same. You have your eye color and eye color, but the allele would be different. This one is coding for red eyes. This one's going to code for brown eyes. Looking at the wing length, you have normal wings and the vestigial wings. Looking at body color, this one's going to have a gray body. This one has an ebony body. And then for the antennas, this one's going to be a long antenna. This one's a short. They could be the same. Like you could have long antenna, but it doesn't have to be. Living organisms can reproduce either sexually or asexually. Asexual reproduction: a single organism will produce offspring that are genetically identical to itself. Prokaryotes reproduce asexually through binary fission as an example, while eukaryotes reproduce asexually by mitosis. In sexual reproduction, the offspring that are produced are genetically different from each other and from their parent or parents. Each of the parent will produce its own reproductive cells, in other words, producing the gametes. However, it is necessary for the number of chromosomes in each gamete to be reduced by half. This is accomplished through meiosis. The gametes undergo meiosis as they are formed inside their corresponding sexual organs. The cells that arise from the diploid cells that contain 2 complete sets of chromosomes. After meiosis, each gamete produced is haploid, each containing half the number of chromosomes. During reproduction, the gametes fuse together during fertilization to produce that first new cell, which is known as a zygote. So here's a question for you guys. If you again would stop the video and go through and answer this question. Meiosis is responsible for more than just reproduction. By reducing the number of chromosomes within a cell, it introduces genetic variation into the gametes and the zygotes that it can produce. Genetic variation is not only caused by meiosis. Mutations can also cause genetic variation, and they can occur at any stage of the life cycle. It is due to genetic variation that natural selection has occurred and has produced such a large range of species that are on the planet. In these 2 diagrams, you're looking at actually this first one for a prokaryotic cell, where you're going to have binary fission occurring, where the organism is just going to it's going to split into 2. These 2 organisms would be identical. When you look at mitosis and the cell's going to divide, again, these 2 cells are going to be identical after going through mitosis. Meiosis is going to have genetic variation. Looking at meiosis, meiosis is similar to mitosis. However, meiosis involves 2 divisions, meiosis 1 and meiosis 2. You still follow PMAT, prophase, metaphase, anaphase, telophase. However, when you're talking about meiosis 1, it would be prophase 1, metaphase 1, anaphase 1, telophase 1. And then when you go into meiosis 2, your PMAT would be prophase 2, metaphase 2, anaphase 2, telophase 2. During meiosis, you get 4 non-identical cells. Meiosis 1 is the reduction division. It is where you're going to have the chromosomes resulting in 2 daughter nuclei with half the number of chromosomes as the parent nucleus. In meiosis 2, the division in which the chromosomes behave just like they did in mitosis, each of the haploid nuclei divides again, giving us a total of 4 haploid nuclei. Genetic variation can be accomplished during meiosis due to crossing over or independent assortment of the homologous chromosomes. Independent assortment states that allele pairs separate independently during the formation of gametes. Crossing over occurs during prophase 1, and it's when these sister chromatids from the homologous chromosomes, they overlap with each other. They're tightly packed together, allowing them to overlap one another, and it allows them to exchange their genetic material. Prophase 1, chromosomes condense and become visible. Chromosomes have already been copied, so each one is made up of 2 identical sister chromatids. The chromosomes arrange themselves in homologous pairs. You can see the homologous pairs arranging together. That's what's known as a synapsis. Centrioles migrate to opposite ends of the cell and form spindle fibers made of microtubules. The nuclear envelope breaks down and the nucleolus disappears. Each pair of homologous chromosomes is called a bivalent. The chromosomes in a bivalent are very close. This is representing that bivalent. Chromosomes together, the homologous pairs, they're very close. A chromatid of one chromosome intertwines with another chromosome, which is what's showing you in this third diagram and also down here if you pay attention not only to the colors but the letters. Excuse me. The spot where the chromosomes cross is called a chiasmata. This is going to allow for crossing over. And so the sister chromatids, those are very close together. And if you look at the diagram, again, the point where they're crossing is known as a chiasmata. They actually are going to exchange genetic information. So when you look at them after the crossing over has occurred, you'll notice that what was originally all blue now has some of that red color, and the one that was originally all red now has some of the blue. They literally have exchanged genetic material. Metaphase 1, spindle fibers attach at the centromeres. The bivalents move to the equator of the cell. Homologous chromosomes in each bivalent remain attached at the chiasmata. In anaphase 1, the spindle fibers begin to pull on the centromeres. The 2 whole chromosomes in each bivalent are pulled apart. Centromeres remain intact. In telophase 1, the chromosomes arrive at opposite ends of the dividing cell. Spindle fibers break down. In most animals, a nuclear envelope forms around each set and the nucleolus reforms. Does not necessarily occur in plant cells. A reduction division has occurred. Each new cell contains 1 complete set of chromosomes that are haploid. This is followed by cytokinesis, which is going to split them into 2. Each of those 2 cells are going to be haploid number of chromosomes. This is representing meiosis 2, where you have prophase 2, metaphase 2, anaphase 2, and telophase 2. This second division of meiosis, which is very similar to mitosis. The overall result is 4 haploid cells from 1 diploid cell. The cells will vary in their genetics. This just again a visual for you to see the first phase of meiosis, looking at PMAT1. And then meiosis 2, PMAT2, where we end up with 4 haploid cells in the end. Okay, question time for you. If you would please pause the video, take a few moments to answer these questions before proceeding. Looking at crossing over a pair of homologous chromosomes in a parent cell, if crossing over does not happen, then as the chromosomes are separated, there's not any crossing over. You'll notice that your capital E, which is representing a long antenna, capital A, which was representing red eyes, hasn't changed from the start. It remains the same. The lowercase e, representing short antenna, and the lowercase a representing brown eyes, again, hasn't changed from the beginning. But if crossing over does occur, then you'll notice as you're comparing these, notice that our color has been switched. We now have red and blue together on both of the chromosomes. And in this case, we started with a long antenna, but with red eyes, but now I have a long antenna with brown eyes. We started with the short antenna with brown eyes, but now I have a short antenna with red eyes. This again is going to lead to genetic diversity depending on which one of these chromosomes is involved with the reproduction of forming a zygote. Looking at independent assortment, during metaphase 1, the orientation or the arrangement, it's random. The production of different combinations of alleles in the daughter cells as a result of the random alignment of the bivalents during metaphase 1. There are actually 8,324,608 different possibilities. This comes from looking at 2N, where N is the haploid number of chromosomes, then factor in all the different alleles of all the different genes on all of these chromosomes. Again, humans have 23 pairs of chromosomes. Comparing independent assortment with crossing over. Again, as you guys are looking at these, pay attention to the colors and the sizes of the chromosomes to see the different possibilities for the independent assortment. As you get 4 different possibilities for your daughter cells, again, different combinations, it depends on which one of these gametes is involved in reproduction is going to determine the genetics for that organism. In crossing over, again, they're exchanging genetic material from the homologous chromosomes, which again is going to get transferred down into those gametes. Looking at gametogenesis, which is the production of sperm cells or egg cells, this is showing you where that independent assortment, it's important. It's showing you how the production of those 4 sperm cells, they're all going to have some differences in their genetics. So if you were to follow through with the meiosis, each of those 4 sperm cells, they ended up with different chromosomes. And then it depends on which one of the sperm cells is involved with the fertilization. It's going to determine the genetics. Same thing is true when you look at the eggs. Now in producing eggs, you're going to get 1 egg cell and then 3 polar bodies. That 1 egg cell, again, depending on how the homologous chromosomes arrange themselves. And then you can also take into account what happens if crossing over also occurs. This is a nice chart that just summarizes the genetic variation, looking at crossing over and independent assortment, and then which one of those gametes are involved with reproduction. Taking a look at gametogenesis in humans. Meiosis occurs as gametes are formed inside the testes and ovaries. The formation of male gametes is known as spermatogenesis. The formation of female gametes is known as oogenesis. Sperm production takes place inside tubules in the testes. Diploid cells divide by mitosis to provide numerous diploid spermatogonia, which grow to form diploid primary spermatocytes. The first division of meiosis then takes place, forming 2 haploid secondary spermatocytes. The second division of meiosis then produces haploid spermatids, which mature into haploid spermatozoas. Oogenesis follows a similar pattern, but many fewer gametes are made than during spermogenesis. This process takes longer with long waiting stages. Oogenesis takes place in the ovaries. Diploid cells divide by mitosis to produce many oogonia. These begin to divide by meiosis, but stop when they reach prophase 1. At this stage, they are called primary oocytes, which are still diploid. All of this happens before a baby girl is born, and at birth, she has around 400,000 primary oocytes in her ovaries. But what happens after this waiting period and she reaches puberty? When the female reaches puberty, some of the primary oocytes get a little further with their division by meiosis. They proceed from prophase 1 to the end of the first meiotic division, forming 2 haploid cells. The division is uneven at this point. One cell gets most of the cytoplasm and becomes a secondary oocyte, while the other is a little more than a nucleus and is called a polar body. The polar body can be thought of as a way of getting rid of half the chromosomes and has no further role to play in the reproduction. Each month after puberty and during ovulation, one secondary oocyte is released from the oviduct from one of the ovaries. If it is fertilized, it continues its division by meiosis and can now be called an ovum. The chromosomes of the spermatozoon and the ovum join together to form a single diploid nucleus called a zygote. The zygote can now divide by mitosis to form an embryo and then a fetus. Looking at gametogenesis in flowering plants, looking at this figure, male gametes are produced within the anthers, and the female gametes are produced in the ovules. The anther, remember, this is the anther, it's attached to a filament. The filament supports the anther, and this is where the male gametes will be produced. The ovules are within the ovary. The ovary is part of the carpel. You had the sticky stigma, style, ovary, and then the ovules are inside. Looking inside the anther, diploid pollen mother cells divide by meiosis to form 4 haploid cells. The nuclei of each of these haploid cells then divide by mitosis, but the cell itself doesn't divide. Cytokinesis does not take place. The result in cells that each contain 2 haploid nuclei. These cells will mature into pollen grains, each surrounded by a protective wall made up of a tough exine and a thinner entyme. One of the haploid nuclei is called the tube nucleus, and the other is the degenerative nucleus for male gametes. Inside the ovule, a large diploid spore mother cell develops inside the ovule. The cell divides by meiosis to produce 4 haploid cells. All but one of these degenerates, and the one surviving haploid cell develops into an embryonic sac. The embryo sac grows larger. Its haploid nucleus divides by mitosis 3 times, forming 8 haploid nuclei. One of these becomes the female gamete.